Hello, welcome to Chapter 6 and talking about viruses. We're going to hang out with Baby Yoda tonight and talk about viruses. So, viruses are really cool and they're all scary. And as we're in the middle of a pandemic right now, as I'm recording this, COVID-19 is like a hot topic, right? So, it's going to be interesting to learn about this and then to be able to apply it to kind of our daily life right now. Okay. So, um, the search for the elusive virus. Louis Pasteur postulated, which is just a fancy word for thought, right? He thought that rabies was caused by a virus, and this was way back in 1884. And then Ivanovsky and Bejernik showed a disease in tobacco was caused by a virus, and this again was in the 1890s, so 100 and I guess 30 years ago, right? And in 1950, virology was a multifaceted discipline, and this is what virology is to study viruses. And so, what are viruses? Well, viruses are non-cellular particles with a definite size, shape, and chemical composition. So, what is the position of viruses in the whole biologic spectrum? Well, there's no universal agreement on how and when viruses originated, and viruses are considered to be the most abundant microbes on Earth, which is kind of scary. And viruses also played a role in the evolution of bacteria, archaea, and eukarya, and viruses are obligate intercellular parasites. So the thing that makes viruses different than other things is they can't reproduce on their own, right? They have to take over a host, and that would be what's the obligate intercellular parasite, because they're taking over the other host and like hijacking that host cell so that it can make copies of the little virus. Okay, and that's kind of why they're scary. So the general size range for most viruses are most of them are under zero point 0.2 microns. And so you remember when we're looking at things on our microscope, things are, we can blow it up to about 100x. But these are so small that they actually need a special microscope, and this is called an electron microscope. So electron microscopy is very cool because you can see a lot of different details. And so this would be like a couple pictures of, different things. So we have Staphylococcus, which we've looked at underneath the microscope already in the lab, right? So we have the pox virus, the herpes simplex virus, rabies, HIV, influenza, the adenovirus, T2 bacteriophage, all the way down to, a hemoglobin molecule, which is, a blood cell, right, which is about 15 nanometers. And our E. coli is only 2 microns, is big actually, and it's about a 2 micron long molecule, where again the strep is about 1 micron. So you can see that viruses are really small. Okay, so a viral structure, the viruses bear no resemblance to cells, and they lack protein synthesizing machinery. And to synthesize is just a fancy word to make. Okay, so the reason why viruses have to take over and hijack a cell is because they can't make their own proteins. So they have to hijack the cell in order to have them make the viral proteins. And viruses contain only the parts that are needed to invade and control the host cell. So the virus particle has a covering and a central core. The covering is a capsid, and this is an envelope. And this envelope isn't found in all viruses, right? And then you also have your nucleic acid molecules, which is either DNA or RNA. Then you have your matrix proteins and enzymes, which are not found in all viruses. But again, you don't see any replication things here in the central core. So the general structure of viruses are, we have those capsids, and these are where all viruses have capsids, and those capsids are protein coats that enclose and protect the nucleic acid. The capsid together with the nucleic acid is called the nucleocapsid. And you guys have known so far that I am very big on root words, right? So nucleo would be nucleus, capsid is that capsoid. So just kind of break it down and combine it when we have to come up with new words, okay? And some viruses have an external covering, and this is called an envelope, kind of like we put an envelope on the outside of our mail, right? So they have an envelope, and those that are lacking an envelope are called naked. And each capsid is made of identical protein subunits, and these are called capsometers, which make up facets or sides. Okay, so if we're talking about a multifaceted problem, this is multiple-sided problems. So again, another fancy word, but it just means side. Okay. There are 2 structural capsid types. The first one would be a helical, and this contains a helix, which is kind of like the twisting ladder. And so a continuous helix of capsomers, which forms a cylindrical or a circular nucleocapsid. Okay, so you can see that here. The other one is called an icosahedral, and this is 20-sided with 12 corners. So that looks pretty darn complex, eh? But icosahedral, okay? So 20 sides with 12 corners. Um, so looking specifically at the viral envelope, um, these are mostly found in animal viruses, and they're acquired when the virus leaves the host cell. And these are exposed proteins on the outside of the envelope, and these are called spikes. And these spikes are essential for attachment of the virus to the host cell, because remember, they have to go in there, attach really, really hard so that they can take over that host cell. So those spikes are kind of going to be like claws going in. So what is the function of this envelope in viruses? Well, this is to protect the nucleic acid when the virus is outside of the host cell. And this also will help the virus bind to the cell surface, and it assists in the penetration of the viral DNA or RNA into a suitable host cell. We also have complex viruses. Okay, so they have to be fancy, right? And these would be atypical. So these aren't your normal viruses, but we do have some tricky ones, okay? So the pox virus, this lacks a typical capsid, and these are covered by a dense layer of lipoproteins. And some of them have bacteriophages, and these have polyhedral nucleocapsids along with that helical tail and attachment fibers. So let's look at that a little more closely. So we can have that capsid head that contains that nucleic acid. Then we have a collar, rest in peace Ruth Bader Ginsburg with her little lace collar, right? We have a collar, and then we have the sheath, and then we have the tail pins and the base plate, right? The tail fibers. So you can see that some of them have a little bit more advanced structures. So the types of viruses are summarized here, where we have those complex viruses, we have enveloped viruses, we have the non-enveloped virus, naked viruses. Okay, and some examples of the complex would be the pox, right? Some of the enveloped would be, you know, these guys, right? With the, I can't really read this very well. I'm sorry. So this is a figure that's in your book. Okay, but anyway, these are the types, and it's a very good summary for you. Okay. So when talking about the nucleic acids of viruses, we have a viral genome, okay? And this is either DNA or RNA, but never both, okay? It can have one or the other, where us as humans, we have both, right? Our DNA codes for RNA, and then from that, we can go on and make proteins, ouch, okay? But these nucleic acids, they carry the genes that are necessary to invade those host cells and then redirect that host cell's activity to make new viruses. Like I said, they kind of hijack it, right? And then the number of genes varies for each types of virus, from a few up to hundreds, right? So it just depends on the severity of hijacking and the type of virus and what they need. So DNA viruses usually use double-stranded DNA, but they also could be single-stranded. And so double-stranded is abbreviated DS, single-stranded is SS. And this DNA can either be circular or linear. Okay, so a circle or a line. For the RNA viruses, usually are single-stranded, but again, they can be double-stranded. So it's kind of flip-flop. DNA is usually double-stranded, RNA is usually single-stranded, but there are exceptions to those rules. And these may be segmented into separate RNA pieces. So single-stranded RNA genomes ready for immediate translation are called positive sense RNA, whereas single-stranded RNA genomes that must be converted into a proper form are called negative sense RNA. So the general structure is that we have preformed enzymes that can be present. These would be polymerases, replicases, or transcriptonases. Okay, fun fact, any enzymes, well, to back up, enzymes are proteins, and proteins are functional parts of our body, right? So enzymes are specialized proteins, and it's cool because enzymes end in this suffix of ase. So if it ever has ASE, then you know that that's going to be an enzyme, which is, again, also a protein. So the polymerase is going to be an enzyme that deals with DNA or RNA. Replicase replicates, it makes a copy of RNA. Where reverse transcriptase, this synthesizes or makes the DNA from RNA. And this one would be important in, for instance, the AIDS virus. But again, remember that proteins end in ACE, ASE, and those are going to be specialized workers of the body. Okay. So how do we classify viruses? Well, the main criteria presently used are structure, the chemical composition, and the genetic makeup. So we currently recognize 3 orders, 63 families, and 263 genera of viruses. And the family name ends in viridae. So for example, Herpesviridae. So where the genus name is going to end in virus. So simplex virus. And so then we have the family and the genus would be the herpes simplex virus 1, okay? And that would be in the family Herpesviridae, and then the genus of simplex virus. So how do viruses live? Like, how do they multiply, okay? Well, the general phases in animal virus multiplication cycle are these following 6. So the first one is adsorption. And so this would be the binding of the virus to the specific molecule on the host cell. Then we have penetration, and this is when the genome enters into the host cells. Then we have uncoating, and this is where the viral nucleic acid is released from the capsid, or the outer covering, right? Then we have synthesis, fancy word for making. So this is where the viral components are produced or made. Then we have assembly, okay, kind of like an assembly line. This is when new viral products are going to be constructed. And then we have release. And this is where the assembled viruses are now going to be released by the budding or exocytosis or cell lysis. Okay, so basically it takes over the cell. I kind of think of it as like a plunger, where it's just like you put like a little needle in and then you plunge it in. Okay, so that's going to be the penetration, and then the uncoating, where it's released from the capsid. It does its little thing and then either like engulfs the cell, right? Or it, like through cell lysis, it's going to just explode the whole cell, or it will release that little virus by a bud, and that would be the exocytosis. So here are your little pictures that show, from hijacking into replication into ejecting it from the cell. So what is adsorption and host range? Well, the virus coincidentally collides with a susceptible host cell and absorbs specifically to receptor sites on the membrane. And then the spectrum of cells of a virus can infect, and this is the host range, okay? So an example of hepatitis B would be it finds human liver cells. Poliovirus, this is primate intestinal and nerve cells. And then rabies is various cells in many different types of mammals. Okay, so these are the specialized cells that they can specifically recept to. Okay. So what is penetration and uncoating? Well, the flexible cell membrane is penetrated by the whole virus. Like I said, it's kind of like that needle injecting in there. And then we have endocytosis. And this is when the entire virus is going to be engulfed, brought in, and enclosed in a vacuole or a vesicle, which you can just think of as like a little pocket, right? And then we have fusion. When we fuse something together, if we ever have welded or glued something together, you fuse it together, okay? So fusion is going to be the envelope that merges directly with the membrane that's going to result in that nucleocapsid's entry into that cytoplasm. And so here's a couple different types of penetration and uncoating. You can see the host, the different parts of it coming in, but basically it all ends up with the viral DNA or RNA getting into the host cell and then taking over. So talking about replication and protein production, these vary depending on whether the virus is a DNA or an RNA virus. Okay, DNA viruses generally are replicated and assembled in the nucleus, where RNA viruses are generally are replicated and assembled in the cytoplasm, okay, outside of the nucleus. Well, the RNA viruses, I already said that. So positive sense RNA contains the message for translation, where negative sense RNA must be converted into the positive sense message. Then we have release, okay? And this is when the assembled virus leaves the host in one of 2 ways. Either it buds, which is going to be exocytosis, where that nucleocapsid binds to the membrane and then it pinches it off and sheds the viruses gradually, and the cell is not immediately destroyed. Or we have lysis, and lysis is with the non-enveloped, and this is when the complex viruses are released when the cell dies, and like it explodes, right? It lyses, it blows up, and it just releases all of the viral little guys, okay, viral babies. So this can obviously cause damage to the host cells, and this is, in my opinion, why viruses can be so incredibly scary, because they're taking over and hijacking cells in your body, right? And so we can have these damages, and one would be the cytopathic effects. And so this would be the viral induced damage to the cells. So we can change the cell's size and shape. We can have cytoplasmic inclusion bodies. We can have inclusion bodies. The cells can fuse to form multinucleated cells. We can break apart or lyse that cell, right? We can alter the DNA because we're hijacking it and taking it over to make it for the actual virus too, or we can transform cells into cancerous cells with uncontrolled replication, and that can be really bad, right? So what about persistent infections? Well, persistent infections, this is when the cell harbors the virus and it's not immediately lysed. So the virus isn't going to go in, hijack, take over, do its business, and then boom, it's gone. It holds on, it hangs on for a bit. And this can last for weeks or for the whole entire lifetime. And several of them can periodically reactivate. And this is called the chronic, reoccurring, latent, like lazy state. And so one example would be measles. And this may remain hidden in brain cells for many, many years. Another one is herpes simplex virus. And this is where you have cold sores like on your lips or genital herpes, right? Or you can have the herpes zoster virus, and this is with the chickenpox and shingles. They can reoccur. So what about viral damage? So some animal viruses can enter the host cell and permanently alter its genetic material that results in cancer. And this would be a transformation of the cell. Then the transformed cells have an increased rate of growth and alterations that occur to their chromosomes and the capacity to divide for an indefinite amount of time that can result in tumors. Because what is cancer? It's uncontrolled cell growth, right? So this is basically, it's taking over, right? And then those mammalian viruses are capable of initiating tumors, and these are called oncoviruses. And so one example would be Papillomavirus, which is cervical cancer, or Epstein-Barr virus, which is Burkitt's lymphoma. We also have multiplication cycles in bacteriophages. So what are bacteriophages? Well, we'll break it down. Bacteria would be the bacterial, right? And then phages would be viruses. So the most widely studied are those that infect. Okay, and so one would be, for instance, E. coli. We've already looked at this in the lab. Okay, this is a complex structure and it has DNA. Multiplication goes through similar stages as the animal viruses, and only the nucleic acid enters the cytoplasm. The uncoating is not necessary. And then we release what is a result of cell lysis, and this is induced by the viral enzymes and the accumulation of viruses, and this is called the lytic cycle. So lysis is the actual active word, and lytic is the descriptor. So it would be a lytic cycle. So what are the steps in the phage replication? The first one would be absorption, which is when we bind the virus to a specific molecule on the host cell. The second would be penetration, and this is where that genome is going to inject itself or enter into that host cell. We have replication, and this is where the viral components take over, okay, and they're going to start making their own copies, right? And so they're reproduced. Then you have the assembly, and this is where the viral components are assembled. You have maturation, and this is when those little viruses are going to mature and get ready to go out and do their own little harm. And then you have the lysis and release, and this is when the viruses leave the cells in order to go on and infect other cells. So here's a summary figure of that as well, and you can go ahead and look in your textbook for that picture. So let's compare bacteriophages and animal viruses. So here you have the bacteriophage, and then you have the animal virus, and you can see this chart is going to compare adsorption, penetration, synthesis and assembly, viral persistence, the release from the host cell, as well as cell destruction. Okay, so check out that book or that table in your book. Okay. So lysogeny. This is the silent virus infection. So not all phages complete the lytic cycle. Some DNA phages, which are called temperate phages, undergo absorption and penetration, but they don't replicate. And so this viral genome inserts into a bacterial genome, and it becomes an inactive prophage or before phage, right? And the cell is not broken apart or lysed. The prophage is going to be retained and copied during normal cell division, and this is going to result in the transfer of temperate phage genome into the host cell progeny otherwise known as lysogeny. And an adduction can occur, then this results in the activation of the lysogenic prophage, which is going to be followed by the viral replication and cell lysis. All right, so here is your fun little diagram of the lytic and lysogenic life cycles. And again, this is in your book, so you can look into it a little closer, okay? So lysogeny. Lysogeny results in the spread of the virus without killing the host cell, okay? So these phage genes in the bacterial chromosomes, they can cause the production of toxins or enzymes that can cause the pathology. And this would be the lysogenic conversion. And some examples will be diphtheria, cholera, botulism, right? So all of those microbes below. So what are the different techniques that we use in cultivating and identifying these animal viruses? Well, we have obligate intracellular parasites that require appropriate cells to replicate. And the methods used could be the cell or tissue cultures, and these would be where we have cultured cells that grow in sheets that support the viral replication and permit observation for cytopathic effects, so we can study them. You have bird embryos that we can use. This is where we incubate an egg, which is going to be an ideal system, and then the virus is going to be injected through the shell so that it can take over that really large egg cell. Or you can have live animal inoculation, and this is occasionally used when necessary, but we try not to use animals when we don't have to. But when we do use them, we have to use them for a good reason, and this would be a good example of that. So there are different methods that we said, we can either grow, we can inoculate the egg, and this is how they do it, right? And the inoculation of the embryo through the egg yolk, right? And how you would actually do it under a sterile environment. So what is the medical importance of viruses? Like, why do we care? Well, viruses are the most common cause of acute or very quick onset infections. And there are several billion viral infections this year, especially when we have a pandemic, right? So some viruses have high mortality rates, and there is a possible connection of viruses to chronic afflictions of unknown causes. And viruses are major participants in the Earth's ecosystem as well. So back to that fourth point, the connection of viruses to chronic afflictions. So this is kind of a gray area right now that we're in with COVID, right? With COVID-19. So I'm recording this on 9/24/2020. And right now we're in the middle of the COVID pandemic, right? And so the problem is, it might not be the worst thing to catch, but we don't know the downstream effects. And one of my very good friends is an emergency room physician, and, she's looking at, these x-rays and stuff. And, the people are presenting with lungs that look like TB lungs, right? They look like popcorn lungs with tuberculosis, right? So we don't know the downstream effects yet because this is still a very novel or new virus. Okay, so that's really important to point out. We can also use it for detection and treatment of animal virus infections. So this is more difficult than other agents, but we consider the overall clinical picture. So we would take an appropriate sample, we would infect the cell culture, look for characteristic cytopathic effects, and then screen for parts of the virus, then screen for immune responses to the virus, which would be like antibodies. And then we can have antiviral drugs that can cause serious side effects. So we don't want to go willy-nilly treating everyone that doesn't have it, right? So we just have to treat it for when it's necessary. So what about prions and other infectious particles? Well, prions, these are misfolded proteins, and these contain no nucleic acid, and these are extremely resistant to usual sterilization techniques, and they can cause transmissible spongiform encephalopathies, such as fatal neurodegenerative diseases. Now, back in the 2000s, we had mad cow disease, and this was a prion disease. So before, I personally hadn't heard about them, but now, it's happened in my lifetime now, this pandemic, so I've lived through a couple of fun, interesting things. But anyway, so prions, they were really interesting because, an infected patient would come into the ER, they'd treat them, and then they'd sterilize all of the instruments and stuff like they normally do, but then they found that the next patient got it. So they actually had to study these quickly, and then they had to figure out how to sterilize more appropriately so that those prions weren't getting passed on to the next patient. That was the surgical instruments were used. So for a while, and autoclaves weren't working, right? So they had to come up with a new sterilization or a higher temperature, longer time, or new chemical add-in, whatever. But for a little bit, like a week or two, right, people were getting infected just from using the regular autoclave because it wasn't killing off these prions. So that's kind of an interesting thing that's happened in my life as well. So some of these prion diseases are common, like scrapie in sheep and goats. So basically what happens is they go up and they like lose their balance and they're itchy. And so they go up against a fence and they scrape up alongside of it. So that's why it's called scrappy or scrapie, right? We also have bovine, which is cow, spongiform encephalopathies, oh my God, which would be shortened BSE, and this would be like mad cow's disease. We have wasting disease in elk, and actually in deer too, right? And then in humans, we can also have Creutzfeldt-Jakob syndrome, and this would be one that would be caused by prions. All right, last slide. Other non-cellular infectious agents could be like our satellite viruses, and these would be dependent on other viruses for replication. And an example would be adeno-associated viruses, and this replicates only in cells that are infected with the adenovirus. Or a delta agent, which is a naked strand of RNA that's expressed only in the presence of, for instance, hepatitis B cells. Or you have viroids, and these would be short pieces of RNA with no protein coat that have only been identified in plants. So viruses, sneaky little hijackers. They, for me, are kind of scary, but they are what they are. So, yeah, all right. Thanks for coming, and we will talk to you guys in class. See ya.